Hello, Mr. Allman here, and this is your Allman Cast on DNA replication. As a reminder, the central dogma of biology states that DNA, which is capable of copying itself, is transcribed into mRNA, mediated through the process of translation by tRNA to produce a protein. So the majority of DNA is used to be able to build proteins which are going to do most of the jobs within the cell. Now, because DNA has such a central function in the cell and in the life of the cell, any change to a DNA sequence would change the resulting proteins and thus irreparably harm the function of the cell. Therefore, it's vital that cells have a reliable way to be able to copy DNA prior to the process of cell division. So DNA replication is going to show us how it is that cells accomplish this task. So just like in transcription and translation, we have a beginning, a middle, and an end. And so initiation is the term that we use to describe how it is that DNA replication actually begins. There are specific locations, actual sequences of DNA, those are called origins, and those are at many different points along a chromosome. Not one, but many points on a chromosome. When replication begins, replication actually proceeds in two directions from that origin. You can see in this electron micrograph how it is that the DNA strand has separated into two parts. The pink arrows show the points where the DNA strands have separated, and this creates a circle shape. This is called the replication bubble. And replication is actually extending in both directions from those arrows. Standard level needs to be aware of DNA polymerase as well as helicase. However, higher level should be familiar with all of the enzymes here on this page, which I'll detail now. DNA polymerases. There are many different kinds of these. There are many dozens of polymerases, and even within prokaryotic and eukaryotic cells, within a single species, you can expect to see at least a half dozen polymerases working together, each with a slightly different job in terms of the process of DNA replication. The two that you should be familiar with are DNA polymerase 3. DNA polymerase 3 is the enzyme that is primarily responsible for the majority of the length of the DNA chain. DNA polymerase 3 is known to be able to copy over 1,000 bases per second as it moves along the length of the DNA template and replicates a new strand. DNA polymerase 1 is responsible for replacing the RNA primer after it has been used. A second enzyme I'll mention is helicase. Helicase opens up the helix, which separates the strands from each other. You can see in green here, the helicase is at that point where those two strands separate from each other. Once they're separated, remember that there's a chemical attraction between those complementary bases in those two DNA strands. So in order to allow replication to occur before those strands come back together, single-strand binding proteins, represented here in gray, actually cover up those hydrogen binding sites for those complementary base pairs to prevent the strand from closing up before polymerase can come in. Primase is another enzyme, and primase actually puts down a very small chain of RNA nucleotides that are complementary to the DNA nucleotides on the template strand. The reason for this is because DNA polymerase can only add bases onto an existing chain. So DNA polymerase is not capable of starting a new chain. It's only capable of attaching the correct complementary bases to an existing chain. So primase puts down some initial RNA bases so that the polymerase can come in and add on to them. Finally, DNA gyrase, which is in the image here, it's called topoisomerase, so those are synonyms to each other. DNA gyrase rotates the DNA because as these are being pulled apart, remember that the DNA is a spiral staircase. That double helix, as it separates, as the double helix is unwound, the parts that are still wound up are going to get tighter and tighter. So in order to prevent any torque that's going to cause the DNA molecule to break, the DNA gyrase is going to rotate the DNA to relieve some of that tension. So in this process, nucleotides are going to be added always to the 3' end of the DNA strand. And the nucleotides will be added due to the complementary base pairing rules. So DNA is anti-parallel. So you can see that our original, in our template strand here, our template strand is 3' at the top, 5' at the bottom. We have a new strand being created that adds on bases from the 5 towards the 3' end. So the 3' end here at the bottom of this DNA strand is the place where polymerase is going to pull in and attach a complementary nucleotide. Those free nucleotides are called nucleoside triphosphates because they are nucleotides not just with one phosphate, but with 3 phosphates attached. So as polymerase adds on that base, those 2 phosphates on the end are actually cleaved off, and the energy that is released from those phosphates being hydrolyzed is what's utilized to be able to form that new covalent bond between the existing DNA backbone and the triphosphate, the new nucleotide that's being added on. So now that is how it begins. It begins with the RNA primer adding down bases and DNA polymerase using free nucleotide triphosphates to be able to add on nucleotides that are complementary to the template. Now the problem is that because DNA is anti-parallel and nucleotides can only be added in one direction, it is difficult then for DNA to be added in a continuous way. So if you look at this bubble here, the replication machinery moves along the chromosome. One strand of DNA can be made in a continuous 5' to 3' piece. That's called the leading strand. In our image here, you can see that the dark blue represents the template strand. So this oval overall is called the replication bubble. The origin of replication is present here in the middle of that bubble. So from the middle is where the process of polymerization of a new strand is going to occur by polymerase. So on the bottom, you can see that there is a leading strand which is building a complementary strand continuously in the 5' to 3' direction. On the opposite side, that same thing can happen going in the other direction. So starting from the origin, not moving towards the right, but now moving towards the left. So we have a leading strand moving towards the left. Now to be able to fill in those other parts, we now have a lagging strand to be able to deal with. So we'll take a look at that. Again, you can see here the leading strand, the red represents the primase, the RNA nucleotides that were added by primase. The light blue represents the new strand that is complementary to the template. We have DNA polymerase here at the end. We have as well the helicase, which is unwinding those proteins from each other. So now let's take a look at how the lagging strand is going to be made. The lagging strand is going to be made in a discontinuous fashion. As you can see in this image at the bottom, the overall direction of replication is from right to left. The end result is that we need to make a new DNA strand that's 3' to 5', which is the opposite direction in which polymerase works. Polymerase cannot build a strand from 3 towards the 5' end. Therefore, the polymerase needs to build the new DNA strand in the opposite direction from the overall direction of replication. So instead of being able to move continuously from right to left, the polymerase only works in this image from left to right. And so it will build those segments in the 5' to 3' direction, but in small segments called Okazaki fragments. So it begins with the RNA primer adding down a small bit of RNA onto which the DNA polymerase will be able to add. The polymerase then will add on bases from the 5' towards the 3' direction, in this image going from left to right. Then the polymerase will detach at the end of that fragment. A new RNA primer will be added towards the 3' end of the template, and then allow now a second fragment to be produced by polymerase, again from the 5' to 3' direction, showing from left to right, until it reaches back to the original primer. So now we have a primer, then we have new DNA nucleotides, a primer, DNA nucleotides, and that pattern continues as those fragments are built in a discontinuous way. Then the enzyme ligase is going to come in, it's going to stitch together those pieces of DNA, replace the RNA with DNA, and ensure that the strand, that the backbone of that DNA, that those sugar phosphate backbones will be connected between the fragments. So when we look at this overall, you can see on the top, the origin of replication was here on the right, so the direction of replication on our image is moving towards the left. We have the leading strand being built in the 5' to 3' direction in a continuous way, and we have the lagging strand on the bottom being built in a discontinuous way from the 3' towards the 5' end. Again, with polymerases adding from, in this image, left to right, left to right, left to right, left to right, backing up and moving closer and closer to helicase, which is unwinding the DNA. Helicase is going to continue towards the left until it meets up with another replication fork, because this process that you see here, this bubble, is going to be repeated up and down the length of the chromosome. So it's not just one enzyme that's responsible for being able to produce those new DNA strands along the length, but rather many different enzymes all working simultaneously, copying different parts of the DNA until the whole length of DNA is actually copied. Here is a couple of videos that will be helpful. So if you look at the notes section of the video, you will find the link to the Prezi to be able to access and look at a couple of different videos throughout. Now, termination. Termination is going to be what happens at the end of the elongation phase as the actual polymerase reaches the end of the chromosome. Now, because of the way that polymerase works, by holding on to the DNA and adding on in its active site new nucleotide bases, at the 5' end of the lagging strand, there are 100-200 base pairs that the polymerase is not physically capable of adding on because it detaches from the DNA template. Now, if this continued, every time the DNA was copied, the chromosome would get shorter and shorter after each replication, encroaching upon a gene, and therefore you would lose vital genetic information that's needed by the cell. You can see in the image here that the overhang here represents part of that fragment that's going to be lost. As it's lost, then we have a new leading strand that's going to, as it copies each time, become shorter and shorter with each of the resulting daughters. So as a way to be able to accommodate the fact that polymerase isn't capable of extending all the way to the end of the chromosome, we have the telomere is the name of the eukaryotic chromosome, short repeating sequences of DNA. So the telomere is going to be a sequence that isn't transcribed or translated, therefore losing a little bit of that genetic information is not going to be harmful to the cell. So it is thought that as our cells age, telomerase becomes inactive. As it inactivates, it no longer adds on those extra bases onto the end of the chromosome, so after each division the chromosome becomes shorter, and once the chromosomes reach a critical point and have lost a certain amount of DNA bases at the end, the cells will go through a programmed cell death and they will die. And so aging is somewhat a result of DNA information being lost through the process of DNA replication. Finally, proofreading is an important step that happens at the end of DNA replication. There is an error rate, about 1 in 10,000 mismatched bases. So because of the length, the 3 billion base pairs that we have in our full genome, that would lead to hundreds of thousands of mutations per replication. So we have molecules that proofread DNA. So As you can see, because of the size of the bases, because we're supposed to have one purine and one pyrimidine base in each complementary base pair, anytime that there is a mismatched base pair, there should be a small bulge in the DNA because the bases no longer bind together effectively and they can actually be physically larger in size. So we have nucleases, which are going to perform a DNA repair. They will cut out a section of the DNA that actually has that error in it, and then stitch in a new set of DNA nucleotides that are going to be complementary to the template strand. Then ligase, again, will come in, stitch together the phosphate sugar backbone to complete that repair process. Proofreading is happening continuously along with DNA replication. So as DNA replication is continuing in both directions from the replication bubble, the proofreading enzymes are also going to be following along and ensuring that any of those mismatched bases are removed and replaced. This decreases the error rate to less than 1 in 10 billion. So per cell division, it is less likely than not that you're going to actually have a mutation that has changed one single base within that. However, remember that we have millions of cells, and because we have millions of cells, these mutations are happening. They do accumulate over time. Please look back through the notes. Please also look at the Prezi to watch the videos, and let me know if you have any questions. Thank you for watching.